A dihybrid cross is going to be a cross that looks at the inheritance of two different genes at the same time. So this is really combining two separate monohybrid crosses together. So as I've drawn it here, we have two different flowers, and we're really representing two different flower characteristics. We are going to look at a color gene, and I'm going to represent this with the letter R. And we've got two different versions. We've got red and we've got yellow. And red is going to be the dominant version, so it's going to get the capital R. And the lowercase r is going to be for yellow. The other characteristic that we're going to be looking at is petal shape. And since we can choose any letter we want to represent these genes, I'm just going to pick a random letter here, and I'm going to use the letter A. And the reason I'm using the letter A is because the letter P or S for petal or shape, that would be a poor choice because the capital version looks very similar to the lowercase version. So if you do use letters where the capital and the lowercase versions look very similar, a lot of times you do end up making just careless errors along the way. So I'm going to stick with A here. And so I've got a capital A and a lowercase a. And capital A is going to be the rounded, and lowercase a is going to be pointed petals. Okay, so now we're going to go through and we're going to look at a cross that involves both of these. So up here at the top, these are going to be my parents that I start with. This would be my P generation or my parental generation. So if we go through and we start talking about these individuals, in this case, they are both going to be homozygous individuals. So since they're homozygous, this red curved one or red rounded one, it is going to have two capital R's because it is homozygous for red, and then it's also going to be homozygous for round, so I'm going to give it two capital A's. We want to write the two R's next to each other and the two A's next to each other. For the yellow pointed one, it is homozygous for yellow, which is lowercase r, and it is homozygous for pointed, which will be two lowercase a's. So these are my parents. Now, just as before, if these parents are going to produce offspring, the first thing that has to happen is the process of meiosis. So we have to get egg and sperm. And I'm going to make this my female parent here. And this is going to be my male parent over here. And so when I cross them, in the egg from this parent, we are going to have haploid eggs. And that means that they're going to get one version of the R gene and one version of the A gene. So since we don't really have much in the way of variation here, we've got two big R's, so we're going to get a big R because that's the only possibility, and we're going to get a big A because that is the only possibility for that one. Over here on the sperm side of things, it is a similar situation because, again, there's no variation there. So we've got a lowercase r and a lowercase a. So this makes this first step quite easy, because we're going to just take the two that we get and we're going to combine them together, and that would be through the process of fertilization. And when they combine, we're going to write our two R's next to each other and our two A's next to each other. So this right here, this would be the zygote, and that would also be our F1 generation. Okay, so we've produced that. Now what we're looking at here, the letter combination inside the little circle that I've drawn, that would be the genotype. So 100% of our F1s are going to be heterozygous for R and also heterozygous for A. But then we could also talk about their phenotype. And so we would evaluate the phenotype for the R gene, which means it's going to be red. And then we would evaluate the phenotype for the A gene, which means that it's going to be round. Okay, so this is the phenotype of the F1 generation, and 100% of them are going to be red and round. Now, if we were to go on and produce the F2 generation, we're going to use two F1 individuals to serve as the parents for that. So let's go ahead and take two F1 individuals and let's cross them to get the F2 generation. So here I have two F1 individuals, and I'm going to cross them with each other to get the F2. So I want to emphasize that that is always what we will use to serve as the parents for our F2 generation. So as before, we always have to go through meiosis first. So now when we go through meiosis, it's a little bit more complicated because we do have different versions of R and we have different versions of A as well. And we can actually go through and we can calculate how many possibilities we should come up with when we do form the gametes. So to do that, if we want to calculate our number of possibilities, if we look here and we just look at how many different R's we have, there are 2 different alleles for R here. And then we could also look and see that there are 2 different alleles for the A gene. And so if we just multiply those together, we get 4 total possibilities. So what this means is that there are 4 different gametes or 4 different eggs that we should be able to form from this particular individual. So if we go through and we look at those 4, one way to do this, if I just write this individual a little bit bigger here, is we know that we're going to get 1 R and 1 A in every one of the gametes. So the way that I do this is I look at, we could get a possibility of a capital R and a capital A. We could get a capital R and a lowercase a. We could get, that's the only combinations with the capital R. We could get lowercase r and capital A. Or we could get lowercase r and the lowercase a. Now, one thing about writing our gametes here, keep all of the R's on the same side and keep all of the A's on the same side. And that will just simplify things when you do start to draw the Punnett squares. And also look down at all of these different gametes. And these, again, if this was my female parent, these would all represent possible eggs that we could get. And they would be produced by meiosis. But make sure that you can look down them and you can see that every one of them has 1 and only 1 R and 1 and only 1 A inside of them. Again, that's going to be because they're haploid. If we looked at the other parent over here, it really is the exact same one. So we would expect to get the same possibilities in the sperm from this particular one. So again, we're going to look at the capital R and the capital A. You can have the capital R and the lowercase a, the little r and the big A, and the little r and the little a. Okay, so these would be all of the little sperms that we would have. So remember that it's a random selection of an egg and a random selection of a sperm, and so we don't know which one we're going to get from each. And there is a lot of possibilities that we can get here when we do start combining those. And if you just look at the fact that we've got 4 different eggs and you've got 4 different sperm, this could combine in 16 total ways. So we want to draw a Punnett square now that's going to represent all of those 16 ways that these can actually combine with each other. So I'm going to clear this right here so that we have a little bit more room to draw our Punnett square. Okay, so if I'm going to draw my Punnett square, we put all of one parent's gametes down one side. So I'm going to write all of my eggs down this side. And these are the ones that we came up with previously. And I'm going to write all of the sperm possibilities across the top, which did happen to be the same possibilities. So I'm just going to put those up here. And then what we would do is we're going to just divide this off into a Punnett square, but this time it's going to be much, much larger, okay, because we're going to have really kind of a 4x4. So we've got more boxes, and I'm just going to divide it off like this so that you can see all the different boxes that we have. And now, once we've got this drawn as it is here, we can go through and we can start filling in those boxes. So if we do that, remember that to fill in the boxes, we look directly across and directly above, and then we write that combination in the square. When I do that, I want to make sure that I put the 2 R's next to each other and the 2 A's next to each other. And that's just going to help us to see a little more clearly what we are dealing with. So if we go through and we fill in all of the boxes, again, looking across and looking above, I'm going to take a little bit of time and go through and do that. Okay, so now I have all of my boxes filled in, and what these boxes are representing, if we just write it over here, is all of these individual squares are the possible F2 offspring. Okay, because that is what we were trying to take a look at here. So there are a number of possibilities that we have in here. And besides looking at the genotypes, which is the letter combinations that we're seeing, we could also go through and we could assign the phenotypes to them as well. So remember that as long as they have at least a capital R, they're going to be red. So I'm just going to put a little red dot in all of the ones that have a capital R because they are going to look red. So I'll just quickly go through here and you can see that most of them are going to look red. And then the other boxes that do not have capital R's, those are going to look yellow. Okay, so we can assign the phenotypes for flower color. We could also go through and we could assign the shapes. So if I were to do that, if they have a capital A, they're going to be rounded. Okay, so I'm just going to do like this for rounded. And again, that would go with all of the ones that have at least one capital A. And then the ones that do not have a capital A, they're going to be pointy. So I'm just going to do it like this. That's meant to be pointy. So this is going in all of the boxes that have only lowercase a's inside of them. So if we look at what we get at the bottom and we draw this out, we have some that are going to have red flowers that have rounded petals. We're going to have some that are red flowers with pointed petals. And then we're going to have some yellow flowers with rounded petals. And we'll have some yellow flowers with the pointed petals. Okay, so those are our 4 possibilities that we can get. And which one we get just depends on what the letter combination was. So if we go through here and we count them up, if we count up how many happen to have the red round, and I'm just going to mark them off as we go through, this will be 1, 2, 3, 4, 5, 6, 7, 8, and 9. So notice we had 9 individuals or 9 boxes that had at least one capital R and at least one capital A. So they gave us round red flowers. If we look for the pointed red ones and cross them off, here I've got 1, 2, and 3. So I've got 3 of those. If we look for the yellow rounded, I've got 1, 2, and 3. And then if I look for the yellow pointed, it's just the bottom box right here. So there's 1 of those. So what we've just come up with here on the bottom, this would be the phenotypic ratio. And this is the phenotypic ratio, this 9 to 3 to 3 to 1. It's a phenotypic ratio that's very characteristic of a dihybrid cross F2 generation. Obviously, it takes a bit of time to fill in the Punnett square, but once we do have it filled in, we can see what all the possible genotypes are, and we can see what all of the phenotypes are. Now, if we were to add even more genes to this, if we were to look at 3 genes at a time or 4 genes at a time, you can imagine that the Punnett square is going to continue to get much, much larger. And really anything beyond 16 squares in my mind is unmanageable. So it will be very worthwhile to learn to use probability rules to figure out the outcome of genetic crosses so that we do not have to later go and continue to draw these really large Punnett squares.